Good day, everyone. Our topic for this lesson is about the different biomolecules. The chemistry of life explores the essential molecules and processes that sustain living organisms. It focuses on how biomolecules such as carbohydrates, lipids, proteins, and nucleic acids interact to maintain life functions. These elements that you can see on the right are usually combined to form molecules or compounds that sustain life. Now let's go to water. Water is called the supporter of life. Water is vital for life because it acts as the universal solvent, enabling essential chemical reactions and transport of substances in cells. Its unique properties such as high heat capacity, cohesion, they help regulate temperature and support life processes on Earth. That is mainly because of their molecular structure. Now, because of its molecular structure, plants can transport water and allow some insects to walk on water, as you can see in the picture. Water supports aquatic life because when carbon dioxide dissolves in it, it forms carbonic acid which helps regulate pH and maintain balance in aquatic ecosystems. Its solid form, which is the ice, floats on liquid water, insulating the layers below and allowing organisms to survive even in freezing conditions. Now, substances can also be classified into two. We have the hydrophobic and the hydrophilic. We're very familiar with hydrophobic and hydrophilic. Hydrophilic substances like sodium chloride, the one that you can see in the picture, dissolve easily in water because their charged particles interact with water molecules. While hydrophobic substances, they repel water and do not dissolve. Now let's go to carbon. Carbon is called the backbone of life because it can form four stable covalent bonds, allowing it to form or to build large, complex, and diverse molecules. This versatility enables the formation of essential biomolecules like carbohydrates, lipids, proteins, and nucleic acids that sustain life. Now, carbon is also called the backbone of life because it can form long chains and rings, such as the carbon tails in fats that store energy in living things. The same property allows carbon to make hydrocarbons like gasoline, showing its ability to create biological molecules and fuels. Now, what is ATP? ATP is called agent for energy. Why? Because ATP releases energy when its high energy bond between the second and the third phosphate groups is broken during what we call the process of hydrolysis. This process forms ADP or adenosine diphosphate and a free phosphate, releasing energy that cells use for vital processes like movement, transport, and synthesis. Now, these are the common molecules that are significant in the study of life. We have water molecules, carbon molecules, adenosine triphosphate, and biological molecules. We were able to discuss water molecules, carbon molecules, ATP. Now, let's go to the biological molecules. Okay? So, polymer is like a long chain and its links are called what we call monomers. Monomers are the single building blocks while polymers are the big molecules formed when many of these monomers are joined together. These monomers are joined together by what we call covalent bonds. Now, what you can see in the picture, let's focus on the dehydration synthesis. Dehydration synthesis, as you can see in letter A, is the process where two molecules are joined together by removing a molecule of water, as you can see in the picture. This reaction builds larger molecules such as proteins from amino acids or starch from glucose, which are essential for life functions. Now, carbohydrates. Let's go to carbohydrates. Carbohydrates are organic molecules made of carbon, hydrogen, and oxygen, usually in a ratio of 1:2:1. They are the body's main source of energy or immediate source of energy, providing fuel for cells through sugars like glucose. Carbohydrates also serve as structural components in plants, which is the cellulose, and energy storage in both plants like starch and animals, which is the glycogen. Now, monosaccharides are the simplest form of carbohydrates consisting of single sugar molecule that cannot be broken down into smaller groups or smaller sugars anymore. They serve as quick source of energy and building blocks for more complex carbohydrates. The three common monosaccharides are the glucose, which is the main energy source for cells, fructose or sugars found in fruits, and galactose, which is commonly found in milk. What about disaccharides? From the word itself, disaccharides. Okay, disaccharides are carbohydrates made up of two monosaccharide units joined together through a dehydration synthesis reaction. Remember, we discussed dehydration synthesis reaction earlier by monomers are formed by removing a molecule of water. Now, they provide energy and can be broken down into simpler sugars for use by the body. Now, these are the three common disaccharides. The sucrose, which is glucose plus fructose or the table sugar. We have the lactose or the glucose plus galactose or which is the milk sugar. And maltose, which is glucose plus glucose found in germinating grains or malt sugar. It's also called the malt sugar. Now, polysaccharides, what are polysaccharides? Polysaccharides are large carbohydrates made of many monosaccharides linked together, serving as energy storage or structural support. Take note of that. We have storage polysaccharides and we have the structural polysaccharides. Storage polysaccharides like starch in plants and glycogen in animals store energy that can be broken down when needed. Structural polysaccharides such as cellulose in plant cell walls and chitin in insect exoskeletons provide strength and support instead of storing energy. Now, storage polysaccharides are large carbohydrate molecules that organisms use to keep extra energy for later use. In plants, starch is the main storage polysaccharide found in seeds, roots, and tubers where it provides energy for growth and reproduction. In animals, glycogen serves the same role and is stored mainly in the liver and muscles, ready to be broken down into glucose when the body needs quick energy. Now, for structural polysaccharides, these are carbohydrates that give strength and support to cells and tissues instead of storing energy. In plants, cellulose is the main structural polysaccharide forming strong cell walls that help maintain shape and protect against external forces. In animals, fungi, chitin serves as structural polysaccharide making up insect exoskeletons and fungal cell walls for rigidity and protection. Now, starch. Starch in plant cells, glycogen in muscle cells, and cellulose fibers in plant cell walls are composed entirely of the glucose. Next, starch and cellulose are both made of glucose but their functions differ because of the position of the bonds between glucose units. Now, let's take a look. In starch, the glucose molecules are linked in the same orientation or alpha bonds. Okay, as we can see in the picture, making it helical shape that is easy for enzymes to break down into energy. In cellulose, the glucose molecules are linked in an alternating orientation or the beta bonds, creating straight chains that pack tightly together, making them very strong and suitable for structure. So, what does that imply? If the position changes, the function also changes from energy storage to support or vice versa. Next, structural polysaccharides are strong materials of organisms. Chitin is a carbohydrate used by arthropods like insects, spiders, crustaceans, and other arthropods to build their exoskeleton. Its structure, it's like the cellulose in plants, but the glucose monomer has nitrogen-containing branch. Now let's go to proteins. Proteins are essential biomolecules made of amino acids that play a central role in nearly all of the biological processes that we have. They function in building structures, speeding up chemical reactions, right? Like enzymes, transporting substances, and defending the body. Now, this is the structure of proteins, and we call this one an amino acid. An amino acid is the basic building block of proteins, and each has a central carbon or central carbon atom. We call it the alpha carbon. Attached to this carbon are four groups. We have the amino group, a carboxyl group, a hydrogen atom, and a variable side chain, or what we call the R group, the one on top, the one you can see on top. The R group is what makes each amino acid unique and determines its properties and role in proteins. Now, these are some of the amino acids. These are the nonpolar side chain that are hydrophobic amino acids. As you can see, we have glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. These ones are the polar side chains that are hydrophilic amino acids. So we have serine, threonine, cysteine, tyrosine, asparagine, and glutamine. Now this one, the last set, these are the electrically charged side chains, or we call this one also the hydrophilic amino acids. We have the aspartic acid, glutamic acid, lysine, arginine, and histidine. Now, proteins are made of chains of amino acids that fold into specific shapes to carry out vital functions in the body. Let's have one example here. Glutathione is a small protein. Actually, it's a tripeptide made up of 3 amino acids. We have the glutamate or the glutamic acid, cysteine, and glycine. That works as an antioxidant. It protects cells by neutralizing harmful molecules and helps maintain overall health and balance in the body. That is just one example. But there are a lot more proteins made up of chains of amino acids that folds into specific shapes to carry out vital functions in our body. Okay? Now let's go to the functions of proteins. Okay, we're going to discuss 8 functions of proteins here. First one is enzymatic proteins. There are digestive enzymes that catalyzes the hydrolysis of bonds in food molecules. Proteins also act as defense. We have antibodies, inactivate and help destroy viruses and bacteria in our body. Proteins also functions as storage. Okay, storage of amino acids like the one that you can see in the picture. We have there the ovalbumin. Okay, it also functions as transport of substances. Okay, we have here hemoglobin, the iron-containing protein of vertebrate blood, transports oxygen from lungs to the other parts of the body, together with the other organs in the circulatory system, right? Next, another function of protein, we have the hormonal proteins for coordination of organisms' activities, receptor proteins so that our cells can respond to chemical stimulus, contractile and motor proteins, of course, for the movement, and structural proteins for support. And one example of it is keratin that we can find in hair, right? Okay. Now, glycosidic bonds in carbohydrates are formed when 2 monosaccharides join together through a dehydration reaction. Okay? Take note that we call it glycosidic bond. Okay? It links sugar molecules to build disaccharides or polysaccharides. What about in proteins? Peptide bonds in proteins, on the other hand, form between the amino group of one amino acid and the carboxyl group of another, creating long chains of amino acids that will fold later on into functional proteins. Now, take note of the name of the bonds that joins the biomolecules together. For polysaccharides, it's the glycosidic bond. For proteins, it's the peptide bond. Now, peptide bond proteins are like stitches in knitting, linking the amino acids tightly so the yarn, if you can see in the picture, forms a continuous fabric with a specific shape and function. So we can have an analogy of these bonds of proteins like the ones in knitting. Now, the polypeptide long chains are twisted, it's folded, it's coiled into a molecule of unique shape. So each of the protein has their own unique shape, which will later on determine how the protein will work. Now, proteins fold into unique 3-dimensional shapes based on the sequence of their amino acids. Now, these shapes are very precise because of the specific chemical interactions between the side chains, hydrogen bonds, and other molecular forces. The unique shape of proteins, take note, is what allows each protein to perform its special function, such as binding to molecules, enzymes that speed up chemical reaction, or giving structure to the cells. So that means their their function will depend on how the proteins are folded. Okay? Now, proteins have many functions which is determined by its structure. So we have four structures of proteins. First one is the primary structure or the linear sequence of amino acids, and the secondary structure, which is certain repeating patterns. We have the alpha helix and the beta pleated sheet. Now, the primary protein structure, as you can see in the picture, is the linear sequence of amino acids joined by peptide bonds. Remember that? Peptide bonds, like beads on a string, and this order determines how the protein will fold. Okay? Now, the secondary protein structure, the one that you can see below, is the first level of folding, okay? Where the chains form regular patterns such as alpha helices, or we call it coils. Can you see the alpha helix structure there? Okay? Or the beta pleated sheets, or what we call the folded sheets. Okay? They are held together by hydrogen bonds. In short, the primary structure is about the sequence, while the secondary structure is about the local folding patterns that give the protein its first shapes. Okay? Now, we also have tertiary structure and quaternary structure. Tertiary structure of proteins is the overall 3D shape of polypeptide chain. The quaternary structure applies to protein with more than one polypeptide chain or subunit. Let's take a look at it. Tertiary protein structure is the overall 3D or three-dimensional shape of a single polypeptide chain. Take note of that, okay? Again, it's the overall 3D shape of a single polypeptide chain formed when secondary structures fold further and are stabilized by interactions such as hydrogen bonds, ionic bonds, disulfide bridges, and hydrophobic interactions. While quaternary structure, let's take a look at the quaternary protein structure. It exists only in proteins made of two or more polypeptide chains. Okay, we discussed polypeptides earlier, where these folded chains join together to form functional protein complex. Okay, so as you can see in the picture, there are two polypeptide chains joined together. Now, in short, tertiary structure is the unique 3D shape of one chain, while the quaternary structure is how multiple chains fit and work together. Okay? Now, this image on the left explains the primary structure of proteins, which is the specific sequence of amino acids that make up a polypeptide chain. Let us use insulin as an example. The chain shows how two chains A and B are held together by disulfide bridges, and we can see that there are slight differences in amino acid sequence among humans, bovines, hogs, and sheep, the one that you can see in the table. Now, that table of amino acid sequence for human, bovine, and hog, with the use of that, let us identify the insulin source of the polypeptide chains that we can see on the left. Okay? So since, let's take a look, the 30th amino acid, the one that we can see in chain B, okay, the 30th amino acid is alanine, and the 8th and 9th and 10th amino acid, as we can see in the table, for chain A are alanine, serine, and valine. Therefore, the source of amino acid presented is from the bovine. Okay. Now, what does that imply? What does that imply? Now, what happens when there are slight differences in amino acids? Okay? So, what does the previous slide imply? So, if there are slight differences in amino acid sequence, the primary structure changes. Okay? Again, when there are slight differences in amino acid sequence, the primary structure changes. Now, what happens when the primary structure changes? It can affect how the protein folds into 3D shape. Even small changes may alter the protein's stability, activity, or how it interacts with other molecules. Now, sometimes having little effect, or sometimes causing loss of function or disease, as in genetic mutations. Okay? Now, proteins can fold and align themselves into alpha helix and beta pleated sheet with repeating patterns, and some don't have repeating pattern are called the random coils. Keratin found in hair is an example of an alpha helix protein. And the strong silk fiber spider web is an example of a beta pleated sheet, as you can see in its structure. Now, the 3D arrangement overall shape of a polypeptide resulting from the interactions between the side chains of various amino acids, as I mentioned earlier. Now, quaternary structure, as we discussed, okay, is the overall protein structure, spatial relationship between subunits in a protein that has more than one polypeptide chain, as we discussed earlier. Okay, now the quaternary structure of hemoglobin in red blood cells and the structure of heme. Now, what is the common disease related to red blood cells? Let's take a look. Some people's hemoglobin differs from the normal only in the beta chain and only in one position, which is the glutamic acid in the 6th position. It's replaced with valine. Now, this is known as sickle cell anemia. What does that imply? So, it implies that if there is a slight difference in the position of amino acid, it changes its structure. It changes its protein structure. Okay? Now, collagen is a fibrous protein with 3 identical helical polypeptides intertwined to suit its function as girders of connective tissue in skin, bone, and ligaments. Okay, so we are very familiar with collagen, right? We are using that one for our skincare. Okay? Now, let's go to lipids. Lipids are a diverse group of biomolecules that are generally hydrophobic, meaning they do not mix well with water. They play important roles as long-term energy storage, components of cell membranes, and signaling molecules. Now, common types of lipids include fats, oils, phospholipids, and steroids, each serving different functions in living organisms. Now, fat, let's take a look at the fat. It serves as an energy reserve made up of 2 kinds of smaller molecules, the glycerol and the fatty acid. Now, fats or triglycerides are made up of 1 glycerol molecule attached to 3 fatty acid chains. The glycerol serves as the backbone while the fatty acids extend from it like tails. This structure is efficient for storing energy that organisms can use when needed. Okay? Now, fats can be classified into 2 categories in the context of nutrition. We have the saturated fat and the unsaturated fat. We're very familiar with saturated and unsaturated fat. What's the difference between the two? The difference between saturated and unsaturated fat lies in the presence of double bonds in their carbon chains, right? Saturated fat has no double bonds, meaning all carbon atoms are saturated with hydrogen. This makes their chains straight, so they can pack tightly together and are usually solid at room temperature like butter. Now, for unsaturated fat, on the other hand, has 1 or more double bonds that create bends or kinks in the chain, preventing tight packing and making them usually liquid at room temperature like olive oil. Okay? So, what is the difference? Which one is better to consume? So, butter is made from animal fat and it contains more saturated fatty acids and some natural trans fats which can raise cholesterol if eaten in excess. What about margarine? Margarine is made from vegetable oil, so it has more unsaturated fatty acids, but some types, especially older or cheaper ones, may contain artificial trans fat created during hydrogenation, which are even more harmful to heart health than saturated fats. Now, in terms of health, soft or non-hydrogenated margarine with little to no trans fat is generally better than butter, but moderate consumption still, of course, of either is important for our balanced diet. Now, let's go to phospholipids. We're very familiar with phospholipids because of the phospholipid bilayer, right? Phospholipids are made of glycerol backbone attached to 2 fatty acid tails and 1 phosphate group. The fatty acid tails are hydrophobic, that means they're water-fearing, while the phosphate head, okay, the head of phospholipid is hydrophilic or water-loving. That unique structure, as we discussed from our previous lesson, allows phospholipid to form cell membranes where they arrange into a bilayer with heads facing the water and tails tucked inside or hiding inside. Okay? Now, as cell membrane, they self-assemble into double layer. That's why they are called bilayer or the phospholipid bilayer. Okay? Steroids, they are lipids with a structure made up of 4 fused carbon rings instead of fatty acid chains. Now, different chemical groups attached to these rings will give each steroid its unique function, such as cholesterol, testosterone, and estrogen. We're gonna take a look at that one, how their structure looks like later. Okay? The rigid ring structure makes them important for cell membrane stability and as signaling molecules in the body. Now, these are the examples of steroids that are essential for life. We have cholesterol, estradiol, testosterone, progesterone, cortisol, aldosterone. If you can see, they have different attachment to the 4 carbon rings. Okay, that's what makes them unique from each other. Okay, now, anabolic steroids are synthetic substances similar to male hormone testosterone that promote muscle growth and increase strength. Okay, that's one example of a steroid. Now, athletes get banned from using them because they provide an unfair advantage in performance, and they can cause serious health risks such as liver damage, heart disease, hormonal imbalance, and aggressive behavior. So that is why it is banned. Okay, it's banned from sports or from the organizations, athletic organizations. Okay, so these sport organizations prohibit them to ensure both fairness and athlete safety, of course. Now, every molecule and compound inside our body are working together to make you alive. So, yes, let us love ourselves. Okay? Now, let's go to nucleic acids. Nucleic acids are biomolecules that store and transmit genetic information needed for growth, development, and functioning of all living things. The 2 main types are the DNA, which carries hereditary instructions, and RNA, which helps in, of course, protein synthesis. Okay? Heredity is the process by which traits and characteristics are passed from parents to their offspring. Now, what's the difference between heredity and gene? Okay? A gene is a specific segment of DNA that carries the instructions for making proteins which determine the traits. In short, heredity is the passing on of traits, while genes are the actual units of information that make heredity possible. Now, gene expression is the process by which the information in a gene is used to make a functional product that is usually a protein. Okay? It begins with transcription. Okay, as you can see, transcription, where the DNA sequence of a gene is copied into a messenger RNA or an mRNA. Now, that mRNA then undergoes translation, where ribosomes, as you can see in the cytoplasm in the picture, will read its code to assemble the amino acids into a protein. Now, this process will ensure that genes control traits by directing the production of specific proteins in the cell. Now, 2 kinds of nucleic acids in the cells, as I mentioned earlier, are the RNA or the ribonucleic acid and the DNA or the deoxyribonucleic acid. The polymer is called polynucleotide, and our monomer, which is the building blocks of nucleic acids, we call it a nucleotide. Take note of that. Okay, let's go back. Polynucleotide is the polymer. Nucleotide is the monomer. Okay? Now, what are the differences in components of nucleoside, nucleotide, and polynucleotide? When we say nucleoside is made up of only 2 parts. Take note of that. Nucleoside is made up of only 2 parts. Nitrogenous base attached to a 5-carbon sugar. Please feel free to pause the video so you can follow. Nucleoside is made up of 2 parts. A nitrogenous base attached to a 5-carbon sugar. While nucleotide, take note nucleotide, which is the monomer of nucleic acids, has all 3 components. What are the 3 components? A nitrogenous base, a sugar, and at least 1 phosphate group. Take note of that, okay? You can pause the video for you to follow. Nucleotide has all three components. Phosphate group, the sugar, okay? Or we call it the pentose sugar, sometimes ribose sugar, and the nitrogenous base, okay? What about a polynucleotide? Polynucleotide is a long chain of many nucleotides that are linked together by phosphodiester bonds, forming the structure of DNA and RNA. The one that you can see in the leftmost, the strand there, the one in the picture, the one in the leftmost is the polynucleotide. Okay? Now, two monomers nucleotide is bonded, as I mentioned earlier, by phosphodiester bond. Take note of the bond, okay? Which links the phosphate group of one nucleotide and the sugar of another nucleotide. Again, what is being linked by the phosphodiester bond? The phosphate group of one nucleotide and the sugar, okay? The pentose sugar of another nucleotide. Nitrogenous bases are not part of the backbone as with the side chain of amino acid. Now, the sequence of nitrogenous bases, the adenine, thymine, cytosine, guanine in DNA, forms the genetic code that is unique for each organism and even for each individual cell type. This sequence will determine the order of amino acids in proteins, which gives rise to different structure and functions. While all cells in an organism share the same DNA sequence, different cells read or express only certain genes, making them specialized, like the one in our muscle cells and nerve cells. Because of this unique base sequence and selective expression, every cell has the instructions needed for life but carries out only the functions suited to its role. Now, DNA molecules have two strands of polynucleotide, forming the double helix structure. Okay? So, it is in an anti-parallel orientation or complementary orientation. Again, take note ha, the structure of nucleic acids is double helix in an anti-parallel orientation. Okay? Now, the two strands of DNA are linked together by hydrogen bonds. The two strands, okay, phosphodiester bond is different from hydrogen bond. Take note of that, okay? Two strands of DNA are linked together by hydrogen bonds between their nitrogenous bases. Adenine will always pair with thymine through two hydrogen bonds, while cytosine or C pairs with guanine through 3 hydrogen bonds. Now, these specific base pairings hold the strands together in a complementary way, forming the stable double helical structure of the DNA. Now, unlike DNA, RNA is usually single-stranded, so it does not form a permanent double helix. However, bases with the same RNA strand can fold back and pair with each other through hydrogen bonds also, creating loops and hairpin structures. This folding gives RNA its unique shapes, which are important for its roles in protein synthesis like mRNA, tRNA, rRNA, and the regulation of cellular processes. So they are able to fold also to form different functions that are essential for protein synthesis. Now for our summary, living organisms are composed of 4 types of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Now, polysaccharides, proteins, and nucleic acids are polymers constructed when the particular type of monomer forms long chains. Lipids are varied and do not have particular monomer.